Kashna?
Yes. One moment, madam.
Okay.
Start, madam. Start the class, madam.
Okay, Sharada madam.
Okay, madam. Okay.
You can start.
Good morning, all. This is Sharada, PGT in Bioscience, TSRJC, Kodakandala, Girls. Welcome you all to my online class. In first year Zoology, the second unit is Structural Organization in Animals. We have completed up to a level of organization, symmetry, coelom, and animal tissues. That is epithelium and connective tissues, some part. Today we will discuss the fluid tissue of connective tissue that is known as the blood. So let's start our class. Okay. So recapitulation of yesterday's topic. Yesterday we have discussed about the compact bone and the compact bone parts. Let us recall them once again. In a compact bone, two main parts are there. Just like they are epiphysis and diaphysis. The two extended, expanded epiphyseal plates, in between these two epiphyseal plates, epiphyseal regions, diaphysis is the region which is long in structure. Okay. Between the epi and diaphysis, we are able to see the metaphase region. Okay. The new terms of yesterday are periosteum and endosteum. Peri means the fibrous connective tissue which is covering the bone part, which is outer side, is known as the periosteum, and endosteum means which is covering the marrow cavity. So in between these two layers, the matrix which is laid in the form of lamellae is divided in like a outer circumferential lamellae and inner circumferential lamellae. And here we are observing the round structures which are known as the Haversian system or osteons, are surrounded just a concentric manner. Those are called as the concentric lamellae. Between the two Haversian systems, the lamellae portion is known as the interstitial lamellae. In the Haversian system, we are able to see the concentric lamellae which are having the osteocytes in lacunae portion, and the a minute cilia-like structures are known as canaliculi, and all these combinedly called as like Haversian canal and concentric lamellae, lacunae and canaliculi, totally called as the Haversian system or osteon. And in a Haversian system, the Haversian canal is having a connection of an artery and a vein and lymphatic vessel, and it is having the link with the marrow cavity with the Oakman's canal. Okay, so these are the parts yesterday we have discussed. I hope you understood. So let's go for the next topic. Okay. The classification of the connective tissue. So here we completed up to bone. The connective tissue remaining portion is the fluid connective tissue. So usually the connective tissue is divided into connective tissue proper and skeletal tissue and fluid connective tissue. So already we have completed the connective tissue proper and skeletal tissue, that is cartilage and bone. And today we are going to learn about the fluid connective tissue, that is the blood. Okay. So let us go for the next slide. Now, just now only we have discussed that the blood is the fluid connective tissue. connective means which is able to connect with the other parts and other organs in our body. Blood. The blood include erythrocytes or RBCs and leukocytes, WBCs and platelets. The process of blood cells is called as the hemopoiesis. So here we are able to see the red blood cells and platelets and white blood cells. So all these are the cells which are present in the plasma fluid. Okay. So this is about the composition of the blood. So it is called as a fluid connective tissue, which is present in the form of fluid nature and consisting with the cells like erythrocytes means RBCs and leukocytes means WBCs and platelets. Okay. And which are present in the plasma fluid. Now, let us go for one by one. Here, in the earliest stages of embryogenesis, blood cells are formed from the yolk sac mesoderm. Later on, liver and spleen serves as temporary hemopoietic tissues. Hemopoietic tissues means, so at the time of embryogenesis, from the yolk sac mesoderm, the blood cells are able to produce. Later onwards, liver and spleen. But after the birth, the red bone marrow is the primary site of hemopoiesis. Okay. Here, the red bone marrow is the primary site of hemopoiesis. The RBCs, erythrocytes of mammals, are circular in shape. How they look like? Circular in shape, biconcave. So here you are able to see a pit-like structure, that is a cave means a biconcave, and anucleate means without having the nucleus. The biconcave shape provide a large surface area. Here the biconcave shape provides a large surface area for exchange of gases. These are 7.8 mm in diameter. The number of RBCs per cubic mm of blood is about 5 millions in man and 4.5 millions in woman. A decrease in the number of erythrocytes is called as a erythrocytopenia, and it leads to the anemia. Usually we heard the term anemic condition means lack of blood cells. So decrease in the number of erythrocytes is called as erythrocytopenia, and it leads to the anemia. An abnormal rise in the RBC count is called as a polycythemia. Got it? So these are the some regarding points of RBCs. So once again, we go for the points. In the early embryonic stages, the production of blood cells takes place from the yolks of mesoderm. Later onwards, liver and spleen. And after birth, a red bone marrow is the primary site of hemopoiesis. Erythrocytes of mammals are circular and biconcave in shape, and anucleate means without having the nucleus. The biconcave shape provides a large surface area to increase the surface area of exchange of gases. So these are 7.8 mm in diameter. The number of RBCs per cubic mm of blood is 5 millions in man and 4.5 millions in woman. Decrease in the number of erythrocytes is called erythrocytopenia. That leads to anemia. An abnormal rise in the RBC count is called polycythemia. Okay. Now go for the next slide. Here you are able to see hemoglobin pigment which give red color to the blood cells, and which is very, very important for the carrying of oxygen to the cells or tissues, and as well as it is able to carry out the carbon dioxide also. Okay. Now let us go for the points. Shortage of oxygen stimulate the kidneys to secrete a hormone called erythropoietin. So usually if in our body the oxygen level is less, at the time the kidneys secrete a hormone that is called as the erythropoietin into the blood. The erythropoietin stimulate the bone marrow. So at that time the erythropoietin stimulate the bone marrow to increase the production of RBCs. So here in this pic you are able to see the iron molecules. Hemo means iron, globulin means protein. So this is the structure of the hemoglobin which give red color to the blood, and it is very, very essential for the transport of gases like oxygen or carbon dioxide. Vitamin B12 and folic acid are required for maturation of RBCs. For the preparation of RBCs, there is a need of vitamin B12 and folic acid. The lifetime of RBCs in humans is about 120 days. The worn out RBCs are destroyed in the spleen and liver. Means after the completion of their lifetime, they will be destroyed in the place of spleen and liver. So this is about RBCs. Okay. Now, WBC, white blood corpuscles. So here WBC means colorless. White blood means they are not having the color. Due to the presence of hemoglobin, RBCs are red in color. Due to the lack of that pigment, WBCs are white in color, or otherwise colorless. They are spherical or irregular in shape and are capable of exhibiting amoeboid movements. The total leukocyte count is 6,000 to 10,000 per cubic mm of blood under normal conditions. The process of formation of WBCs is called leukopoiesis. So here we are observing the white blood cells: neutrophil, eosinophil, basophil, monocyte, and lymphocyte. Okay, I repeat once again. WBC means the colorless cells, otherwise white blood corpuscles. They are spherical in shape, and the total leukocyte count is 6,000 to 10,000 per cubic mm of blood under normal conditions. The process of formation of WBC is called as a leukopoiesis. So here these are the types of the WBC cells which are present in our blood. Now let us go for the detail. Here, the slight increase in the WBC count is during infection and allergy. Usually when we are having any infection in our body, at the time the WBC count will be increased in our body. So that indicates that we are having some infection at any place in our body. And an abnormal increase in the WBC number is indicating a type of cancer called leukemia. If it is increased a lot, so at that time that is called as a type of cancer that is called as a leukemia. Okay. Fall in WBC count is called as a leukocytopenia. If it is increased, leukemia. If it is decreased, leukocytopenia. Here, the WBCs are two types: granulocytes and agranulocytes. So the terms itself, they are indicating if the granules are present, they are the granulocytes. If granules are absent in them, they are agranulocytes. First, let us go for the granulocytes. They contain cytoplasmic granules. Granulocytes contain cytoplasmic granules. Nucleus of the granulocytes divided into lobes. So here you are able to see a granule-like structure. And usually in case of granulocytes, the nucleus is divided into lobes. Means, for example, lobe means just like. So the nucleus will be divided into lobes based on the staining properties. So how they are capturing the stain, according to that point, we are grouping them into three types: eosinophils or acidophils, basophils, and neutrophils. What are the three types? Eosinophils or acidophils, basophils and neutrophils. So here in this picture you are observing basophil, eosinophil and neutrophil. So these are the granulocytes. In all these three types of cells you are observing the granules, and the nucleus is having the lobe-like structure. So these are the common features of the granulocytes. Cytoplasmic granules are present. Nucleus is divided into lobes, and they are staining with some chemicals according to the staining. They are divided into three types. Those are eosinophils, basophils, and neutrophils. Now let us go for the detail. The first one, basophil. Base. Nucleus is divided into irregular lobes. Here, when we are observing the picture, the nucleus is not having any particular shape. Irregular lobes means they are not we can't able to describe them in a particular manner. They constitute about 0.4% of total leukocytes. Cytoplasmic granules are fewer in number and irregular in shape. The granules also are not having certain shape. So here the nucleus is also not having particular shape, and as well as the granules are also not having. Irregular in shape means they are not having a particular shape. They take basic stains. They produce heparin and histamine. In the tissues, just now recently only we completed. Heparin means it is a product which can control the clotting of the blood in the blood vessels. They supplement the functions of the mast cells when needed. So usually the mast cells produce the heparin. So here the basophils supplement the functions of the mast cells when there is a need. Okay, so this is about the basophils. Now coming to know about the eosinophils or acidophils. They constitute about 2.3% of total leukocytes. Nucleus is bilobed. How the nucleus is looking? Bilobed means here this is the one lobe and this is the second lobe. But in case of basophils, we are not able to divide them. It is irregular in shape. And in eosinophils, they stain with acidophils, acidic stain. They are gaining the acidic stain. They play a role in allergic conditions, allergic reactions. Their number increase during the allergic reactions. So when you are suffering with any allergy, at the time the eosinophils number will be increased in your body. They remove antigen antibody complexes. Antigen means any pathogen. Antibody means the substance which is produced by the blood cells. So here they remove the antigen antibody complexes. So this is about the eosinophils or acidophils. Okay, got it? Basophil means they stain the basic they take the basic stains. Acidophils means they take the acidic stains. So especially for allergic reactions, acidophils, basophils means they produce the heparin. The main feature of the basophil is they produce the heparin, and as well as they supplement the function of the mast cells when needed. So these are the points regarding the basophils and eosinophils. Now coming to the neutrophils. Neutral means balanced. Here they constitute about 62% of the total leukocytes. Nucleus is mainly 2 to 5 lobes. Maximum the cells which are present in neutrophils are having either 2 lobes or 5 lobes or 3 lobes. Here in this picture you are observing 3 lobes. They stain with the neutral dyes. They take the stain of a neutral. These are active phagocytic cells, commonly described as microscopic policemen of the body. Because phagocytic means they engulf the foreign particle, and that is the reason they are giving the protection to our body, just like the defense mechanism. And we are describing them as the microscopic policemen. Okay, especially how can we recognize the neutrophil means the nucleus is having the 2 to 5 lobes means here in this picture you are observing the 3 lobes means definitely it is a neutrophil. They stain with the neutral dyes and active phagocytic cells. So they are described as a microscopic policemen. Now, so that is about the granulocytes, means the cells which are composed with the granules in their cytoplasm. Now coming to know about the agranulocytes. Agranulocytes, A means without, granulocytes means granules are absent in them. Nucleus of cells is not divided into lobes. Here the nucleus is not divided into lobes. Simply it is present, but it is not having any lobe-like structure. These are of 2 types. One is the lymphocyte and second one is the monocyte. Lympho and monocytes. Agranulocytes, without having the granules in the cytoplasm. Nucleus of these cells is not divided into lobes. And these are 2 types, lymphocyte and monocyte. Now coming to know about the lymphocyte. Lymphocytes, they constitute about 30% of the total leukocytes. They are small. So when we are observing the WBC cells, lymphocytes are very small in structure. Spherical cells with a large spherical nucleus. When we are observing the cell, the entire maximum portion is covering with the nucleus. And scanty peripheral cytoplasm. Cytoplasm is present but less amount due to the occupancy of the nucleus is it is big in size and it occupied a lot of space. So that is the reason we are observing the scanty peripheral cytoplasm. There are functionally 2 types, B lymphocytes and T lymphocytes. B lymphocytes produce antibodies. So we heard this B lymphocytes number of times. For the production of antibodies, there is a need of B lymphocytes. And T lymphocytes, which also have an important role in immunological reactions of the body. So both the T and B lymphocytes are useful for our body to maintain the immunity. B lymphocytes produce the antibodies and T lymphocytes play important role in the immunological reactions. So this is about the lymphocytes. Got it? Lymphocytes constitute about 30% of the total leukocytes. They're small, spherical, and nucleus is big in size. Cytoplasm is lesser quantity. Functionally, there are 2 types, B lymphocytes and T lymphocytes. B lymphocytes produce antibodies and T lymphocytes are useful for the immunological reactions of the body. Okay. So this is about lymphocytes. Now go for the monocytes. Monocytes are constitute about 5.3% of the leukocytes. The nucleus is kidney shape. How the nucleus is looking like? It is having the kidney shape. These are the largest phagocytes. They engulf bacteria. They differentiated into macrophages when they enter into the connective tissue. So when monocytes enter into the connective tissue, they are differentiated as the macrophages to control the entry of the pathogens or antigens. So this is about the monocytes. So they constitute about 5.3% of the leukocytes. Nucleus is kidney shape, largest phagocytes engulfing the bacteria. They differentiated into macrophages when they enter the connective tissue. Especially, how can we recognize means having the kidney shaped nucleus. These are having large size when compared with the other leukocytes. Okay, so these are the points regarding the monocytes. Now coming to the RBC is completed, WBC is completed. Now go for the platelets. The platelets. So number of times we heard platelets, which are very useful for the clotting of the blood. These are colorless, non-nucleated, round or oval and biconvex. This is the shape of convex. Number of platelets for cubic mm of blood is about 2,50,000 to 4,50,000. They produce in the bone marrow. The average lifespan of platelet is about 5 to 9 days. They secrete thromboplastin and play an important role in blood clotting. So with the help of thromboplastin, there is a chance of clotting of blood. So that is the reason the platelets are useful for the clotting of the blood. It is very, very essential when injury or any damage is occurred any part. So this is about the platelets. So here you are observing the convex shape of platelet. Okay. Now go for the next slide. Here, so already we have discussed the types of blood cells like RBC and WBC and platelets and plasma. So in the plasma fluid, all these are present. So already we have discussed RBCs and WBCs and platelets and plasma. So all these three types of cells are present in the plasma fluid. So this is the composition of the blood. Already we have discussed. Okay. Now go for the next slide. So that is about the fluid connective tissue. So in animal tissue topic, epithelial is one type of tissue and connective tissue is another type. And the third type is the muscular tissue and fourth one is the nervous tissue. So already we have completed the connective tissue topic. So let us go for the muscular tissue. Muscular tissue is mesoderm in origin. It is of three types: skeletal muscles, smooth muscles, and cardiac muscles. The skeletal muscles. First we go with the skeletal muscles. The skeletal muscles, otherwise called as voluntary. Here in this picture, you are observing only striated muscles or skeletal muscles. They are called as the striated or stripped muscles. Long cylindrical and unbranched multinucleated cell. Here in this picture, you are observing unbranched, stripped, multinucleated cell. Skeletal muscle usually work under the conscious control of an organism. Means they work in our control. With our conscious only they can work. Contracts quickly and undergoes fatigue quickly. They contract quickly and as well as they go for the fatigue also very quickly. They are always attached to bones to bring movements of the body. For the movement of hands and legs, usually skeletal muscles attached to the hands and legs. So while with the help of these muscles only, there is a movement of hands and legs in our body. So this is about the structure of the skeletal muscles. Totally three types. How their origin means by the mesoderm, and they are skeletal, smooth, and cardiac. When we go for the skeletal muscles, unbranched, multinucleated strips. You are observing the strips means such a type of strips you are observing. Long cylindrical, unbranched, contracts quickly and undergoes fatigue quickly. They are always attached to bones to bring movements of the body. Okay. Now go for the next slide. Smooth muscles. Second type of muscles are smooth muscles, otherwise called as unstripped. Here we are not able to see any strips-like structure. Otherwise called as unstripped or involuntary or visceral muscles. So why we are giving the term visceral means usually visceral organs means the organs which are present in the abdomen cavity, which are large in size, are known as the visceral organs. Means they are very useful for the functioning of the visceral organs. That is the reason we are calling them also visceral muscles. Spindle shape. So this is the spindle shape. and do not work under the conscious control, and so they are called involuntary, which are not in our control. When they are working, it is not in our control. So that is the reason we are calling them as the involuntary muscles. Smooth muscle exhibit slow and prolonged contractions. Contractions here also takes place, but very slow. They remain contracted for a long period without fatigue. Even they contracted for a long period, but they may not go for the fatigue. But in case of straighted muscles, they contract quickly, and as well as they go for the fatigue also very quickly. Okay. They help in internal movement to regulate daily metabolic activities like digestion and maturation. So all these are the daily like activities which are not in our control. They are going to happen in our daily life. Okay. So that is the reason these are called as the involuntary muscles. Smooth muscles which are not having the strips, and spindle shape. What is the shape of them? Spindle shape, uninucleated. Uni means only one nucleus you are observing. And they are not, their work under conscious and control of so here they are not in our control. When they are working, the functioning of this such type of muscles is not in our control. That is the reason we are calling them as the involuntary muscles. Smooth muscles exhibit slow and prolonged contractions. Even they contract for a long time without fatigue. So usually they are helping the internal movements to regulate the daily metabolic activities like digestion and maturation. Okay. Now go for the next slide. Cardiac. The third type of muscles is the cardiac muscles, otherwise called as stripped involuntary. So here when we are observing the structure, it is also having the strips, but function is involuntary. Means this is a combination of straighted, non-straighted. Short cylindrical, mononuclear or binucleated cells, whose ends which branches. Here you are able to see the branches. Cardiac muscles are involuntary in function. They are not in our control. Contractions are very fast. Cardiac muscle is highly resistant to fatigue. So for example, in our body, if our heart muscle is going for fatigue, so there is no chapter of us, means we can't able to lie. So this is about the cardiac muscles. So cardiac muscle is highly resistant to fatigue. It is very, very important. Contractions are very fast, but are highly resistant to fatigue. These are the muscles which are having the both the natures of straighted muscles as well as non-straighted. Structurally with the straighted muscles, functionally with the non-straighted muscles. This is about cardiac muscles. So the another type of tissue, epithelial is over, connective tissue is over, and muscular tissue is also over. Now coming to know about the nervous tissue. It is very, very important. Without nervous tissue, there is no use of any organ system. They are not able to function without coordination of the nervous system or nervous tissue. Let us go for this topic. Nervous tissue is derived from the ectoderm. So it is very, very important. It exerts the greatest control over the body's responsiveness to the changing external and internal environment. Means, so with the help of the nervous system only, our body can respond for the stimuli which are coming from the external side, otherwise internal. It consists of two types of cells. Neurons means conducting cells. Neuroglia, supporting cells. First go for the neurons. Already you learned in the 10th class, 9th class, number of times about neuron structure. Neuron usually consists of cell body with one to many dendrites and a single axon. Cell body is called as cyton. It contains abundant granular cytoplasm and a large spherical nucleus. The cytoplasm has nissel bodies. Okay, once again I am repeating here. Nervous tissue is derived from the ectoderm. From where does it is deriving? Means ectoderm. It exerts the greatest control over the body's responsiveness to the changing external and internal environment. Means with the help of the nervous system only, the body can respond for the external stimuli and some changes which are going to take part internally also. It consists of two types of cells. Neurons means conducting cells and neuroglia means supporting cells. Neuron. A neuron is usually consisting of a cell body with many dendrites and single axon. Cell body, it is called as cyton, otherwise it is called as cyton. It contains abundant granular cytoplasm and large spherical nucleus, and cytoplasm has nissel bodies. So let us go for the diagram. Here in this picture, we are able to see the structure of a neuron. This is the cell body and this is the axon. A dendrite which is very long in size is called as axon. In the cell body, you are able to see the nissel granules and nucleus and number of branches which are known as the dendrites. And the long portion which is known as the axon. And the terminal end of the axon is known as the synaptic knobs. Let us go for the points. Dendrites. Several short branched processes which arise from the cyton are called dendrites. Several short processes which are arising from the cell body are called as the dendrites. They also contain Nissel's body. So here in this cytoplasm, you are observing the Nissel's granules. So in case of dendrites also, you are able to observe the Nissel bodies. They conduct nerve impulses towards the cell body. So here the dendrites conduct the nerve impulse towards the cell body. Axon. Axon is a single long process that originate from a region of cyton. The cytoplasm of axon is called as axoplasm, which contain neurofibrils, but Nissel bodies are absent in axoplasm. Axon give rise to branches into many fine filaments. So here at the end of the axon, you are able to see many filament-like structures called axon terminals, which end in bulb-like structure. So here you are able to see it just a round-like structure known as the synaptic knobs. They contain chemicals called as neurotransmitters. Okay, so this is the structure of the neuron. Actually, the nerve impulse from one neuron to another neuron will be entered from this way, from the point of synaptic knob. It is able to attach with the another nerve cells, another neuron's dendritic portion. So here the gap place between these two means one neuron's synaptic knob place and another neuron's dendritic portion is called as a synapse or synapse. So this is about the neuron structure. Especially in the neuron, there are two main parts are there. That is the cell body and axon. Cell body is consisting with a large nucleus in the cytoplasm, having number of branches which are known as the dendrites. One branch is very long in size, that is called as the axon. And in the cytoplasm, it is containing Nissal's granules. The Nissal granules also present in the dendrites also. But in the axon portion, the cytoplasm of the axon is called as the axoplasm, which is contained only neurofibrils. Nissal's bodies are absent here. The end part of the axon is called as the axon terminal, which is having fine filament-like structures and with bulb-like structure. They are called as the synaptic knobs. They contain a chemical that is known as the neurotransmitters. So which can supply the message from one neuron to the another neuron. So they attach the gap place between these two portions is called as the synapse. Okay. So this is about the neuron structure. Now, according to the structure, we are grouping the neurons into three types. They are unipolar, bipolar, and multipolar neurons. What are they? Unipolar, bipolar, and multipolar neurons. First, go with the unipolar. In unipolar, so here in this picture, they are sensory neurons having single process arise from the cell body, which divide into two branches. So here from the cell body, a single branch is there, which is divided into two branches. Single process is divided into two processes. That is known as a unipolar. I repeat the definition once again. They are sensory neurons, okay? And having a single process. So here you are observing the process of the cell body is only one here. And arising from the cell body, which divide single process, but two branches are arised. And in that, one is the dendrite portion, another one is the axon portion. That is a unipolar type of neuron. Now coming to know about the bipolar. So this is the bipolar. In bipolar, have one axon and one dendrite directly arise from the cyton. From the cyton itself, one is the dendrite portion, one is the axon portion. Bi means two. One is the dendrite portion and another one is the axon portion. So this is about the bipolar neuron. Now coming to the third one, that is the multipolar neurons. So here in this picture, this is the multipolar. Have one axon and two or more dendrites. Here in case of the multipolar neuron, you are observing only one axon, but the dendrites are multi. So this is about the multipolar neuron structure. According to their structure, we are grouping the neurons into three types. One is the uni, second is the bipolar, and third is the multipolar. Uni means from the cell body, process is single but dividing into two branches. One is the axon and one is the dendrite. Bipolar means from the cell body itself, two branches directly arised. One is the dendrite portion, another one is the axon. Multi means having only one axon but number of dendrites. So these are the types of the neurons. Got it? Now go for what we have learned. So today we have discussed about the blood topic. According to the blood, so here the points plasma and blood cells. In the plasma, the blood cells are present, RBC and platelets and WBC. In case of WBC, granulocytes and agranulocytes. What are the granulocytes? Eosinophils, basophils, neutrophils. And what are the agranulocytes? Lymphocytes and monocytes. So all these topics we have discussed today. Now let us go for the entire recapitulation of the unit. So in the structural organization of animals topic, here we are grouping the animals based upon their structure. that is organ level of organization and symmetry, coelom, and their tissue structure also. We are discussing up to their tissues, which type of tissues they are having, and what are the types of animal tissues. All these we discussed in this lesson. I hope you all understood this one. Now let us go for the assignment. First question. Give an account of the formed elements of the blood. I repeat question once again. Give an account of the formed elements of blood. Compare and contrast the three types of muscular tissues. Compare and contrast the three types of muscular tissues. Describe the structure of a multipolar neuron. Describe the structure of a multipolar neuron. Thank you.